Okay, hey, welcome to Science with Folly, episode number 1. Today we are recapping protein, oh, you can't see it there, protein synthesis. What is that all about? Synthesis means to make some stuff. You know that, don't you? You're not a stupid. Protein, well, that's the stuff we're making. So we're making protein. What's the whole point of protein synthesis? Well, you need proteins for pretty much everything in your body, like the color of your eyes, like I guess the color of your eyes are to do with protein, but everything else is to do with protein too. So what do? What happens? Well, to start off with, you have this stuff called DNA inside your cell nucleus, inside every single cell in your body. The DNA contains a code, and that code codes for a whole lot of proteins. You got it. Then the proteins, how do they get from that code to being actually made? Well, here we have it. If we look at this DNA has got heaps and heaps of genes, and those genes code for individual proteins. Let's put this page over. So here is a gene. A gene is a double strand of DNA composed of a whole lot of nucleotides, and every nucleotide has on the end of it a nitrogenous base, and that base forms the code A, T, G, C, C, T, A, etc., etc. Three of those is called a triplet. A triplet is complemented by a codon in messenger RNA. A, remember, is complementary, becomes U because RNA doesn't have T, it has U instead. T is complemented by A, G is complemented, it's meant to be a C, it's complemented there. And in this case, these three here on the messenger RNA are called a codon. And the codons, remember, code for stuff. What do they code for? Amino acids. That's right, they code for amino acids. You should have known that. Those amino acids, what happens? Where do they come from? What's going on there? So we get messenger RNA copies the DNA inside the nucleus, and this process is called transcription. Then the messenger RNA goes outside the nucleus to the ribosome, and inside the ribosome a process called translation occurs. And translation is where the messenger RNA gets turned into amino acid, proteins. A whole lot of amino acids are coded for by the codons, and we get a big sequence of amino acids joined together by peptide bonds. Then we get a polypeptide. That polypeptide gets folded up into a functional protein. More than one polypeptide chain joined together becomes a big protein. There you have it. If you don't know what I'm talking about, read your book.